Okay, sorry.
You're just in time, Jane. Okay, we can start. Jane is here. So, are you done introducing me?
Yep.
Great, okay. Excellent, we have no more administrative duties. So, thanks everybody for showing up. I wouldn't normally expect any reasonable person to listen to me for an hour, 2 days in a row. That's kind of exorbitant. Initially I thought I might give as a second lecture a talk, like a more traditional recent science result talk about some work that my group's been doing on aromatic carbon in primitive objects that sample the early solar system. And then somehow in conversation with Glennis, we decided, no, let's like open it up. Let's have a much broader discussion about the context of what is happening in the field that has tasked itself with the study of prebiotic chemistry generally, the organic chemistry that led to the emergence of life, the search for life. So this is a set of loosely defined disciplines that are sort of hard to put your finger on what they are, but there's definitely a cast of characters that do them, okay? And there has been for a long time. And I would say that this subject area, life's origins, life's distribution, it has long been in a strange space between definitely good scientific questions, like life clearly emerged, okay? So it had an origin, and it would be important for us to understand that. And there either is or isn't life somewhere else in the universe other than on Earth. That's like a knowable thing. So of course we should be asking this question, and yet somehow when we start to do it, a lot of nonsense crops up. And a lot of really loosey-goosey science and science-adjacent silliness, and just a lot of funny things happen when people begin to do this. And I think that this is both, it's good to understand why that's occurring, what are the failure modes of this community, but also to recognize that's a trait of a particularly exciting class of problems. Ask yourself, how much silliness was there in our understanding of the cosmos beyond the solar system in the year like 1900 or something, or 1850, okay? People thought a lot of weird things, and they were guessing. And when people guess, we do a bad job. Like, we're not actually good at science. Science is good at science. We're not that good at it, okay? And so people do all kinds of strange things when they're faced with disciplines that are ill-defined, that have not yet undergone their transformative discoveries that frame how they interpret things. But to be able to do that, that's like the dream of virtually every working scientist, that you would get to participate in the framing of how things basically work. So in my field of geology, plate tectonics is our framing idea. It's fun for me that I have plate tectonics as a thing I can lean on in doing geology. Even better would have been to participate in the discovery of plate tectonics. So this is why the problem is exciting. In addition to transforming human understanding of itself, to discover its origins, to discover the distribution of life. Of course, yeah, that's all important, but just more narrowly as scientists, imagine that you actually got to figure out something real about this. So this makes it a good subject to talk about, but a problematic one. A second reason to talk about this subject now is we really have entered a new era in it. And it's an era that's defined by a few major groups of discoveries, the most visible of them being the existence of extrasolar planets, the characterization of them. This clearly is catalyzing a huge amount of activity in this area. And there's a feedback that a lot of the work on extrasolar planets is framed by the search for evidence of habitability of life, things like that. And there are other disciplines too that are less visible, they're kind of working a little bit more under the waterline, but they're also making major progress. And the key thing is, these are drawing in communities who are just much more serious and have more targeted, goal-driven, discovery-driven modalities of work. And so everyone around them is just getting better, getting less goofy, I would say, in their approach to these questions. And that's why this moment right now is just different than it was a decade or 2 or 3 ago. It's an excellent time to be working on this problem. Okay, so What I'd like to do is organize this sort of historically. Walk you through, not like the whole history of human thought about this or anything like that, but what is the modern history? And this is naturally going to be very editorialized. It's my perception of what are important things that have gone on. Some of it you know better than I do. Some of it I know better than you do. But this is, I can already tell, this is a group that's happy to ask questions, engage in dialectics and things. So just like jump up and shout if you have something to contribute. Okay, so first of all, I think it's important to understand we have these two questions. How did life emerge on the Earth? And is there life somewhere else in the universe? And can we see it? These are two very different kinds of questions structurally. So the search for life elsewhere has a concrete answer, okay? It either is or it isn't. We have a search algorithm. We define in advance what we think will count as evidence. And then we look for it. Either you see it or you don't. And so it calls for a certain class of things, very clearly defined targets, technological advances that let you see those targets. You have to be persistent, and you have to take no for an answer, okay? If you don't find that evidence, well then maybe the thing isn't there, right? I understand the philosophy of like the lack of evidence isn't the evidence of lack or something, but you have to understand when you have not seen something. And so in this sense, it's sort of like, you know, the search for the Northwest Passage or the search for the Fountain of Youth. Okay, that's one where the answer was no. And I would say this sort of thought process is behind SETI, okay, but it's also behind some of the evidence for biomarkers in atmospheres of extrasolar planets. So it's part of both the somewhat goofier end of the practitioners and the people who are more serious are doing these activities. The study of the origins of life is a very different thing. So this is an event that there's no question that it happened. Like here we are, it definitely happened. There was a time when the Earth was a giant ball of magma, more or less, and then there's life. So something happened in between. What was it? So you don't have this uncertainty that you have in the search for life beyond the Earth, but on the other hand, you will never actually observe in real time all of the details of what occurred. You are reconstructing it after the fact. It doesn't matter how close you get to describing that, you are always working at it, refining it, explicating. And so it's sort of an endless task that just burrows deeper and deeper and deeper into different levels of detail. The most important thing to bring to this is a mindset focused on falsification. So you don't end up in this do loop of confirmation bias. And this field is filled with confirmation bias. I have a pet idea, everything I see somehow is a reflection of my idea, and I just say that over and over and over again until I retire. And that's a modality of work that has been common in this field. And so how do we get out of that? Like, who is showing us a way of breaking that and actually falsifying hypotheses? Detailed explication, you have to be able to go as many layers deep as you can in describing exactly how your hypothesis works. There has to be constraints, and you'd have to have a way of working that says, well, once I have an answer, I keep going. I make the answer deeper, I make it better. And in that sense, it's more like, say, archaeology, where I find a weird pile of stones and I have a thought about what it is. They say this is a temple, like a Bronze Age temple. Where is it? On Malta? Maybe it's a temple. Maybe it was a temple. I don't know. Maybe who knows what they were doing? I wasn't there. So you're guessing, and they're informed guesses, but you're always there after the fact, trying to figure it out. Okay, so evidence for extraterrestrial life. There are two basic forms that it could take. One is indisputable. Like if we go to Mars and we come back with a rock, and the rock looks like that, it's game over. Like there was life on Mars. And there is no disputing it. No reasonable person could dispute it. If they find DNA, which they won't, but if they did, game over. That's absolutely evidence of life on Mars. But more likely, you have this sort of evidence, interpretation of indirect sorts of clues. Maybe instead of looking like a turtle, you find a thing that looks like a booger. And then what is it? Well, one person looks at it and they're like, it's a living booger. And another person's like, it's nothing. It's just like a smear of carbon in a rock. So it doesn't mean anything. There are molecular versions of that, okay? Maybe I find a molecule and I say, well, that's a molecule that's in my cells. It's alive. Well, there's a lot of molecules in your cells. There's like all kinds of things. So meteorites that have organic molecules in them, they have 10 times or more the diversity of organic molecules than as compared to biomass. So there's much, much more diversity in the organic chemistry of non-living systems than there are in living systems. You're going to find your subset of chemistries everywhere. So how do you recognize them for what they are? And then simple molecules, oxygen, methane, phosphine, which is one we'll come back to. These are supposed biosignatures, but they're even simpler than the molecules that are abundant in your body. And so how do you recognize them for what they are? These are the kinds of evidence we need to wrestle with. Like the odds that you find that, that's just a gamble. This evidence is everywhere, but can you interpret it? And so you need to have nuanced, that is complex, but interpretable ways of describing these things, confident understanding of what this evidence is that you're looking at, and the evidence that you're looking at has to be somehow subject to development. You have to somehow be able to test your interpretations. So this other side of the problem, the study of life's origins, it has been approached in two different ways. And one of them approaches it from the top down, where you basically begin with an observation of the living biosphere and its environment and its characteristics, its ecology, all the different sort of evolved things, and you go deeper, 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 until you reconstruct, maybe through genomics, some description of the earliest clear evidence you can find for a living thing from which we're descended. Unfortunately, like that's just the last thing that is a common ancestor to all of us. What was its ancestor, okay? And what was that thing's ancestor? How do you go deeper? How do you get all the way back to the beginning? The second approach is you start at the beginning. You say, well, things like carbon monoxide and cyanide and so forth that are useful chemical precursors for biomolecules in non-living environments, those things exist almost ubiquitously, and I can study the chemistry of how they're assembled into more complicated things, juicy things that are important in metabolism, maybe really interesting juicy things like peptides and components of RNA and DNA. And then what? And then what happens next? How do I take those things and assemble them into molecules that in living systems can only form with enzymatic assistance? So somehow I have to have enzymes to make the things that are needed to make enzymes. Like how do I even get started? And then synthetic biology questions. How do I take those and force them to interact with each other in ways that are required for living cells? So bridging this gap, it feels almost impossible when you approach it without some systematic methodology. Okay, so now let's get into history mode. How do we get to the way this field works right now? Of course, you can go very, very far back in different sort of philosophizing people have had about these questions. I would say the earliest point that really has direct relevance to the way we operate today is the decades immediately preceding the opening of the space age in the mid-1950s. And in this period, say 1850 to 1950 or something like that, one theme that is really surprisingly strong in people's comments and research in this era is they're definitely engaged in this kind of scientific endeavor, but with a prejudice that you are going to find intelligent life very nearby, okay? It was a widespread belief that intelligent life was on Mars, Venus, the moon. This is not just like crazy comic book stuff. This is practicing scientists. And so this leads to the confirmation bias that is very familiar to us from the Percival Lowell's redrawings of Schiaparelli's maps of Mars into these elaborate things. That persisted. Okay, this is a map from the US Army Air Corps from 1962. It has Percival Lowell's ridiculous straight lines of canals and things on it. So this is not just some fringe belief system from the 1800s. And then the roots of SETI go back to this era as well. Nikola Tesla was using his radar antenna and he in fact thought he had positive evidence of received intelligent communications. This is a newspaper headline from 1924 where everybody in America was asked to not do anything with their radio for blocks of 5 minutes at a time so that we could hear communications from Mars because it was in opposition. It was like some sort of fortuitous time to listen. So this is a very widespread belief system, and it is the belief system in which this next generation of scientists grew up. So they have this, their mind is open to possibilities that today seem ludicrous to us. So what are the sort of signature things that emerged from the dawn of the space age up until sort of 1995? This whole 40-year period I would characterize as being a sort of creative chaos. There's no well-defined specialists. There's no clear affiliation of this kind of science with one discipline versus another. There are no governmental organizations that are specifically funding this kind of research at a significant scale. It's just sort of happening. People are doing it out of personal interest, and they're making up their own way. Part of this has permanent value. It is the science that grew out of robotic exploration of the solar system, the Mariner program, Pioneer, Voyager program. So all of those are really motivated and mostly focused on descriptive planetary science. They can happen completely in absence of questions about life's origins and distribution, and they would still have value. This is, in a way, a lot like the forerunner of the current volume of work on extrasolar planetary astronomy. You can totally remove, in fact, maybe it would even help, remove all the questions about the distribution of life and just look and describe what you see, and this is the thing that has lasting value. But the people doing this, they were really thinking about the search for life and the existence of life elsewhere. It was strongly motivating for them, and this is why we get things like the golden record, that's sent off in Voyager 2 and so forth. So this is also the era where sort of amateurish, ad hoc listening for communications, radio communications from intelligent life, turned into a search algorithm. The launching of SETI as an organized activity. The organization itself came into being in the 80s, but Project Ozma in 1960, this is something I think Frank Drake was organized. This is where they're using devices that are really appropriate for targeted listening for radio signals. This is also the era when the Drake equation emerges, and it's one of these things, I don't know what if you've encountered it or like what your opinion is about it. I've always found the Drake equation to be incredibly frustrating as a piece of science. It's basically an attempt to compute the probability or a number density of detectable civilizations in the Milky Way. Like, okay, this is like a hard problem. Let's start by having something we can compute. So we'll compute it as a function of several things that are observables while you're still, feeling your way forward, like understanding the rate of stellar genesis, fractions of stars that host planets. That's something, of course, that we're studying very actively right now. Numbers of planets in a habitable environment. This is a big factor in extrasolar planetary science. Times 4 numbers that we have absolutely no idea about and won't have any idea about until we have concretely detected life on other planets. So it's completely getting the cart before the horse. And the notion that you could evaluate these 4 numbers, that specify the fraction of planets that have life and the life that evolves to be intelligent and so forth, you have absolutely no idea. You have zero constraints. And so what are you supposed to do with an idea like this? And yet it was somehow strongly motivating. Another characteristic of this era is a sort of blurring of scholarship and public discourse and public outreach. That's like a nice way to put it, probably. But the group of people who were, I would say, the strongest, most visible figures, they're all at Cornell. Carl Sagan, of course, Cosmos fame. Tommy Gold, important astronomer, but also an iconoclastic kind of weirdo in areas of the earth sciences and lunar exploration. So bubbling out ideas that were sort of engineered to draw attention and clearly wrong. And so what do you do with these people, all right? They're like important scientists, they're the people leading, and yet they're spouting off all kinds of crazy things that you know are wrong, and so how do you respond? How do you have a scientific discourse with them in the room? But yeah, here's probably the most famous study. You got a Pulitzer for this thing. So Carl Sagan writes a book in the mid-70s about the emergence of intelligence on the Earth and the rank ordering of intelligence of animals. It's all kind of an interesting question, but like, what do you know about it? You're just like making this stuff up. And so this type of quasi-scholarship has always been cooked in to the pot. Okay, let's look at a few other things that have shaped the nature of what people do in this discipline. How do you actively advance the understanding of the origins of life and its distribution of the places? One is to understand the beginnings of the chemistry. This is the start of the bottom-up study of the chemistry that leads to life. Most famously, the Miller-Urey experiment, where they're doing a spark discharge in a reducing atmosphere and observing the synthesis or the appearance anyway of amino acids. This is often remembered as being an important example of how you learn the chemistry that led to life. It's actually a terrible way to learn that chemistry because it's an unconstrained experiment. It's like, oh, I'm going to throw 800 things into a jar and zap it, and then something appears, and then what? You don't even know what the reactions were. Like, you don't have any idea. A serious synthetic chemist would never do this as an approach to this problem. And yet this is how we do it. So how do you bridge this gap between this modality of studying chemistry and a more serious, mechanistic, sort of dispassionate description of the chemistry? There's also then the discoveries of the existence of a great diversity of life-relevant organic molecules, like amino acids, in primitive solar system objects, like the carbonaceous chondrites. And then changing views of habitability. What are the environments of the Earth, and then maybe other planetary bodies, where life could exist? The discovery of life around hydrothermal vents on the seafloor was transformational in this regard. It demonstrated environments at great pressure and temperature, environments isolated from the surface and the sun. They could be places where life flourished. And then the discovery, basically microbial culturing of samples, fluid samples from deep drill cores, showed that life could perfuse the crust. Initially, people were amazed that they could find culturable organisms from 10 meters deep in the ocean floor. Now we know you can find culturable organisms from like 2 kilometers deep in mines. So life finds its way into the crystalline basement rocks. Out of this, you would think that, okay, that's a great discovery, and it's relevant for thinking about habitability and the nature of what life needs and what it does. Somehow that got turned by one of our Cornell luminaries into a statement of the deep hot biosphere, which said petroleum deposits are actually generated from non-biological processes at depth and then seep up to the surface. Absolutely ludicrous. Like, we find fragments of plants in petroleum. Like, obviously, these are things that are created by the breakdown of biomass from the surface biology. And yet, you can, as an intellectual leader in this discipline, say the exact opposite, and somehow that's like an okay thing. You're giving yourself permission to present science that is really divorced from the process of inductive reasoning or hypothesis testing or things like that. Okay, another important, I would say this is the last enormous investment in the pre-1950s mindset. The assumption there's going to be something alive. It's not going to be that hard to find. All we have to do is look, and we will find living organisms on the surfaces of other planets. It will be relatively straightforward. And this is the life detection experiment from the Viking mission that involved the release to a soil sample of a nutrient mixture that had a carbon-14 label, and then the measurement of carbon-14 coming off that sample as CO2. So concretely, it's a measurement of the oxidation of organic carbon, okay? And absolutely, it was oxidized and the CO2 was detected. The question then is why? The current understanding is it's the presence of oxidants like chlorine in the soil that led to the oxidation. But this is debated still to this day. But I would say it's more interesting as an example of what people thought might work than it is a finding itself. Okay, so the geosciences got drawn into this principally through their study of the remains of life in deep time, right? Looking at ancient rocks. So this is a time scale of the geological record. Today, or actually that would be like 500 million years ago, so today might be here. The oldest rocks we know of are just a little bit younger than 4 billion years ago. And what's being cartoonishly illustrated here, summarized here, is the contrast in carbon-13 content between oxidized carbon in the surface environment, reflected in limestones, and reduced carbon in the surface environment, represented by old organic matter. And this offset of about 3% or 30 per mil in the carbon-13 content resembles the offset generated by photosynthetic carbon fixation today. And so the fact that you observe this difference persisting back into the geological past is taken of evidence that we now have a kind of marker for biosynthesis, something we could apply even more, even earlier in the geological record or in another planetary setting. So a lot was made of this and continues to be made of it. This is a quote that was inspired from Carl Sagan, that was inspired from data of this type. And he says, the time available for the origin of life seems to have been short, a few 100 million years at the most. So he's basically saying the duration of the record that we haven't sampled, that's the most time you have to work with before life has to have been present on the Earth. Right, so he's basically saying life has a high probability of emerging. I don't know, how do you go from, even if this is true, how do I go from, I found one example and it happened fast, therefore it is probable. Says who? You still have one, okay? You have a probability of one. So how do I turn that into some sort of probabilistic statement? A deeper problem is, this is a kind of evidence that lacks nuance. It lacks explication, it lacks uniqueness. These things that we have to have in this kind of biomarker search. This is a plot of various sorts of biological carbon isotope fractionations between oxidized carbon and reduced carbon products. So photosynthetic fixation, various plants that use that, various microbial processes. Those are the signatures that inspire this interpretation. These are the signatures of non-biological carbon reduction processes, Fischer-Tropsch sorts of reactions, the thermodynamic equilibrium at moderate temperature, breakdown of organics, oxidation of organics going the other way, and then reduction in petroleum systems to make methane, various forms of carbon in meteorites. This is always the case. So with or without biology, you are always seeing a preferential retention of carbon-13 in the oxidized form and an exporting of carbon-12 to the reduced form. So basically, even though there's a lot of this kind of work that still gets done, it all says the same thing: reduced carbon formed by some kind of reduction. Like you're not, it's not a biosignature. You are not studying life when you do this. It is, yeah, shoot.
Yeah, I mean, I was just interested in the specific shapes of those.
Yep.
Do they correlate to any
Yeah.
you know, time periods that are relevant for, you know.
To first order, they're just blobs, yeah? But this event and the one above it, that is a positive carbon-13 excursion, they are localized in time as well as, in space as well as time. There are specific stratigraphic sequences that carry these really extreme carbon-13 depletions, and they are believed to be a result of a very specific microbial metabolic process. Yeah. Okay, the takeaway here is not that the Earth was dead 2 billion years ago. That's not the point. The point is, this particular observation doesn't tell you anything. The only reason you know that it was a result of life on the Earth's surface is you already knew that, okay? It is a circular kind of argument. So how do you then export that argument to other places? Okay, so near the end of this period, there was one of the most consequential papers in terms of the funding, the structure, the size of the population of people doing this kind of work. This paper was incredibly influential in this regard. It's a paper that examined a meteorite recovered from the surface of the Antarctic ice sheets, the Allan Hills in Antarctica. And it was demonstrated by a variety of lines of arguments to be a fragment of the crust of Mars that was lofted by an impact a number of millions of years ago, floated around in the inner solar system, landed on the Antarctic ice. In this sample, there are 4 lines of evidence that were argued to point to the existence of life in the Martian crust when this rock first formed about 4 billion years ago. One of them is carbonate minerals that were deposited from water. Okay, this is a process that happens without life, but they argue demonstrates habitability, the presence of water, and temperatures that at which life could exist. Second, microfossils. These are little nanometer scale boogers, okay? So like, what are they? Microfossils, that's the argument. The presence of organic molecules, including, actually exclusively, polycyclic aromatic hydrocarbons. Those of you who study anything about the chemistry in the interstellar medium know, well, those are actually the most abundant large molecules in the universe, so like, why do you think they are evidence of life? Unexamined, okay? They are organic molecules, and they're present. Finally, the existence of biominerals. This is a really small magnetite crystal that is found as an inclusion in these carbonates. Who's to say it's biological in nature? Like if we use our turtle versus booger comparison from the beginning, this doesn't look like a turtle, okay? This is just a crystal. Why is it a biological product? Because it reminds them of a similar crystal from a biological material. The scientific community responded to this as meaning, in the end, not much of anything. You have 4 lines of evidence. None of them are really strongly indicative of biology. The fact that they are all together doesn't mean anything more than if you found them separate. And so this is a non-story. Culturally, this had a huge impact. The response to it was
what are other explanations of the microfossils?
So the microfossils, these are, first of all, the synthetic biologists tell us these are too small to have been fossil remains of a living cell. They have too small of a volume to contain the constituents needed by a living system. They are probably just like chemical precipitates. There's virtually every crystal surface that you could examine at high magnification contains little dissolution reprecipitation imperfections on them. It's not really clear what they are. They're bumps.
Independent of life, is that evidence for water?
This is, yeah, there was water for sure. Yeah, that's right. Okay, the important part of this study is not what it did or didn't discover. It is the response to it. Before we go any further, let me tell you a little bit. So this question of, do I recognize life or don't I recognize life when I look at something in a rock? This is an old problem in geology. And they're referred to as sports of nature, things that you look at and you just have a visceral response that you must be looking at a biological product, a fossil of some sort. Nature is a busy bee. It makes all kinds of shapes of things for all kinds of reasons. And so here are dendrites, crystals that grow on the interfaces of sandstones just by seepage of fluids and precipitation of minerals. This is a non-biological growth in a void space in shale. This is just a funny rock that weathered in a goofy way and now looks like a head. Like, none of this means anything. But human beings look at them and are reminded of shapes that they associate with life. And so Stephen Jay Gould put this well. He says there's an internal psychological barrier to understanding nature as constrained as sensory limitations. Your mind is tricking you into some sort of confirmation bias. So the important thing, though, is that that confirmation bias, that line of reasoning, the excitement around it, the authority imbued to some of the authors who are part of that study, it led to an administrative response. NASA created, really in direct response to this one paper, plus an associated body of work that was connected to it, they created the astrobiology program soon thereafter. And so what is astrobiology? It is the top-down funding of a community to pursue some specific goals in this subject area. And they're specifically aimed at the origin, evolution, and distribution and future of life in the universe. That could cover a lot of things. What they really ended up supporting a lot of, first of all, follow the water. So you said, well, what about the carbonate minerals? Did those still be in water? Yes, they did. And so in a way, this was a wise decision to steer away from things that we didn't really have modalities of study to validate into things that we did. Could we recognize where water is in planetary environments and characterize it? So this became the guiding light in Mars exploration. The associated question of habitability, what are environments that are habitable, that life can exist in? This then overlaps with the study of life that is living in environments we don't normally associate with habitability. They are defining, seeking out, sort of using their own evolutionary pressure to fill all of the spaces that are potentially habitable. In studying the distribution of life, you need to focus on biosignatures. What do we accept as a signature of life, a chemical signature of life? What do we not? And then if we're going to be studying that, we have to be able to contrast life with non-biological synthesis. So these became the areas of greatest focus during the astrobiology era. Just as in this first wave of this field, where the really substantive advances came from robotic exploration of the solar system, this has remained true for the subsequent roughly 20 years or so of active funding of the astrobiology program. The biggest concerted push has been in Mars exploration using orbital imaging, orbital spectroscopy, landers, and rovers, mostly guided by the desire to track water, aqueous processes, aqueous minerals precipitated from water, in the Martian environment. Here's an example of a deltaic deposit in a lake that used to fill a crater. Here are spectra showing the distribution of hydrous minerals and carbonate minerals in bedrock that's exposed in the bottom of the crater. And this was only part of a much broader program of exploration that included outer solar system missions, a comet sample collection mission, which we'll loop back to some of those in a minute. Okay, so this is great, and all of those are permanent advances in our understanding of the geology of Mars and outer solar system bodies. There was also a lot of silliness continuing in this field. Here's a collection of a few studies that were published in this era, around early 2000, looking at Mars using telescopic observations from Earth and reporting porphyrins, vegetation, chlorophyll. All of this is totally unsupportable through the spectroscopy They're just suggestive features. And somehow in the year 2000, this is still an acceptable way of doing work, to associate advanced, complex molecules made by advanced life with spectral features that are non-diagnostic. The pinnacle of this field's loss of control over what constitutes a biosignature and what doesn't, what are the bounds of evidence that are acceptable in this field and what are not, came in a study that was published a decade ago now, examining the chemistry of DNA extracted from microbial organisms from an arsenic-rich lake. And the experiment is, we recover and culture microorganisms, bacteria, extract their DNA, sequence it, cut out one of the sequence bands, and then measure that band for its concentration of arsenic. And the conclusion was, I found arsenic atoms, and it's in a DNA sample. It's DNA made of arsenic, okay? Where the arsenic substitutes for the phosphorus. It's a reasonable first order guess, maybe if you didn't know very much chemistry, but very consequential. It suggests there is a parallel genomics, a genomics based upon arsenic rather than on phosphorus. It's a radical, radical suggestion. So how should you respond to it? The chemist's initial response is to say, we actually know about organic molecules that contain arsenate in them like this. And we know that they fall apart in less than a second in water. It's impossible. And what it must be, and this ended up being proven to be the case, is that when they extracted the DNA, other sulfur-bearing compounds were absorbed as contaminants onto the sample. They never demonstrated that the molecules of DNA had phosphate groups in them. Okay, and so it's just loose chemistry, it's loose evidence. The problem was not that it was wrong. Lots of things go wrong in experiments. People see all kinds of things that they misinterpret. This is just normal science. The problem is the response. How do you react to a discovery like this? You throw a press conference, okay? This is 6 months before acceptance of this paper. There is the most widely publicized press conference that the astrobiology program ever did is about this paper. This individual then gives a TED talk about it. They are given the Time 100 Influencer Prize. Like the whole thing just goes completely nonlinear in a very public way. And then as soon as it's published, it gets blown up by subsequent work because it was obviously wrong. Again, the problem is not that something was incorrectly interpreted in a science study. The problem is the culture of the thing. How are you policing the act of discovery and hypothesis testing and so forth? And this is coming from the top down. This is the administrators who fund it. This is some of the lead PIs in this discipline. Highly, highly problematic. So a bigger problem in the long run is that this field organized itself around a modality of study focused on the development of plausible scenarios. Where could life be found? How could life emerge? And there are a set of them, lakes, hydrothermal vents, so on and so forth. The challenge here is there was not a lot of falsification going on, basically none. And so over 20 years, there was an accumulation of plausibility arguments without any falsification or rejection. And so you end up with a community that can't really make progress. All they can do is find new ways of disagreeing with one another over preferences, okay? But they're not disproving. The hypotheses are not really being posed in ways that are disprovable. Okay, and not surprisingly, a few years after this period, the program basically winds down. So this brings us up to maybe 5 to 10 years prior to today. So what is happening that has really transformed this field and brought it back in a new form? The most obvious one is the search for and discovery of extrasolar planets. And it's been incredibly recent, it's come on really, really strong, and it's obviously directly relevant to both of these disciplines. disciplines, both the search for life and thinking about habitability and the conditions for life. More importantly, it's coming from a community that has its own set of motivations, it has its own set of standards for data, for reporting, for interpretation, and it's just operating in a more serious-minded way. In parallel is a community that's existed for a very long time, but is coming on strong. They've had a great last decade of discoveries and a number of intellectual leaders who've been very influential using radio astronomy to describe the chemistry, including organic chemistry, of protoplanetary disks. And the connection between understanding the chemistry of the disk environment and the habitability of planetary environments is an obvious connection to draw. So this has also been extremely helpful to have that community coming into play. The robotic exploration of the solar system has also reached a new peak in some important ways. Most importantly, we now have 2 missions that have returned specimens from primitive bodies in the outer solar system. This is the first time we've ever had these organic-rich early solar system materials uncontaminated by terrestrial residents. It's really this entire history of thinking about organic chemistry in the non-living early solar system environment has been struggling with the fact that our specimens fell through the atmosphere of the Earth and resided on the surface. Now we finally have things that are not subject to that. This is also a period when we've come to a clear understanding that the icy bodies of the outer solar system, the moons of Jupiter and Saturn, many of them either clearly have subsurface oceans or may have subsurface oceans. So this has expanded the concept of what constitutes habitability. These are objects that are clearly not in the habitable zone with respect to surface water, stability, and yet they have oceans. So they're plausibly habitable. The Cassini mission, near the end of its lifetime, observed mass spectra of organic compounds emitting from plumes from Enceladus, one of these icy moons, showing the connection between either prebiotic chemistry or biochemistry and these subsurface ocean settings. And then finally, the Cassini and Huygens probe missions Titan allowed us to explore a surface environment overwhelmingly dominated by non-biological organic chemistry. So these have really given teeth to our attempts to connect direct observations from planetary exploration missions to the questions of life's origin and distribution. A second community that's much less visible, but I would say equally important, is that the study of the chemistry that transformed simple precursors into sort of pre-biological molecules and then into the complex molecules of life. This field was invaded by, taken over by, serious synthetic chemists. And so replacing these scenario-based experiments, the sort of cook and look experiments, we've seen a new wave of studies that are mapping out mechanistic, specific reaction pathways that show the ability to go from a few simple precursors to the entire family of amino acids, nucleobases, lipids, etc., through mechanistically understood reaction pathways. Those have served as a foundation for learning that some of the really exotic molecules that seem to be almost magical in nature, they're always made by enzymes in life, they are structurally complex, they have complex metabolic jobs. This particular case is coenzyme A, which is really important in our central metabolism. A big piece of this you can just cook up in experiments that start with cyanide and formamide, ammonia. So just a few simple precursors can create these molecules that we have always thought of as being things unique to life. That's important for biosignature studies. It's also important as an insight as to the chemistry that life may have taken advantage of. Why do the enzymes make that molecule? Maybe they found it and liked it and decided, I want to make a lot Another important innovation from the chemists has been the discovery of cyclical chemistries that resemble the TCA cycle in our metabolism, but occur without the intervention of enzymes. They're cyclical chemistries that can happen in a jar and act a lot like metabolism. The recognition of pathways for synthesizing oligomers, that is, chains of molecules that are related to one another but not identical, and non-biological pathways to chiral selection, which is a key element of our biochemistry. So the synthetic chemists have basically rewritten this subject in the last 5 or so years and have brought in a much more serious-minded way of studying. In parallel, there's a development in synthetic biology showing the pathways from very simple molecules like fatty acids that we now know how to synthesize without life to systems that will self-assemble into membranes that resemble cells, chemistries that will non-biologically, without enzymes, allow for cycling of environmental variables that drive the templating and replication that's required for our information molecules, DNA. And then most exciting are experiments that attempt to integrate the chemistry that needs to happen inside a cell with the non-biological synthesis of systems that resemble cells that have membranes in them, such as chemistries that have a small RNA strand, some sort of metabolite, and in a self-sustained, non-manipulated fashion, will draw in a molecule from the environment, transform it, and add it to the cell wall, doing a basic function of life, without life being present. Mars sample return is our first plausible chance that we could actually return from another planet a molecule that could be proven to come from life that's not from the Earth. This just got defunded effectively. That's politics, okay? Like maybe that changes. Yeah, go ahead.
Sorry, I'm just struggling with, if we have all of these pieces of, non-biological chemistry-based, like, functions of life happening, then what kind of divides that from, the definition of life?
You have to pull it all together in one time and place so you can do them together. So we know how to walk into a lab, pull jars off a shelf, and make an amino acid. We don't know how to throw all the things from my jars into a beaker, shake it without control, and have a cell emerge out of that. So having the chemistries be simultaneous, make my amino acids while I make my nucleobases, while I make my fatty acids, so forth. Having those find each other and be encapsulated, and then having the information molecules start to replicate and drive and make catalysts and drive metabolite. It feels like way too many steps to envision making them work all at once.
But do we functionally have all of the steps able to be done separately?
Yeah, almost. And I think this is the important point, is what they're doing now is instead of doing a Hail Mary, swing for the fences, I guess that's a mixed sports metaphor, but instead of hoping that in one cook and look experiment, they can go from a dead jar to something like a cell, they are instead reducing this thing to an engineering problem. Understand every component part, have some hypotheses about orders of operation that need to be enacted, and then in the future, engineer a process that combines those known steps. That's basically the logical path that they are constructing now. But that couldn't be done until they understood the elementary chemistry. And they couldn't understand the elementary chemistry until they studied it in a more serious, controlled way. Yeah. Okay, and even SETI is seeing a resurgence of activity. This thing, what is it, the Robert C. Byrd Green Bank Telescope, I guess it basically was going to be shut down and then received funding from one of the breakthrough initiatives in order to keep it alive and repurpose it for SETI. Okay, so pulling it together, this discipline now has, I would say, a new structure. And it's drawing in communities that were never seriously engaged in it before and bring to it a culture more focused on observational discovery, more focused on serious-minded papers, less focused on speculation, scenario pitching, this sort of thing. And this, I would say, is its basic structure. There is an origins community looking at the processes through which life emerged, and there is an astronomy-driven discipline that is thinking about habitability and the search for life beyond the Earth. The origins community, I would say the strongest parts of it right now are the synthetic chemists and synthetic biologists. And then we have a vestigial sort of rump community of the geologists and people studying life in extreme environments, the geobiologists or astrobiologists, and people studying evolutionary biology. They are the residual from the astrobiology period, and a few of them are hanging around and trying to reintegrate into this new world that's being led by people who have intruded it from chemistry and biology and from the astronomers. The study of life beyond the Earth is, SETI still exists as its own standalone activity, but extrasolar astronomy is clearly where all of the excitement is. Solar system astronomy and robotic space missions, Mars sample return has some possibility, probably the only real possibility in the next 10 or 20 years of handing you a sample where you go, whoa, this thing definitely is a residue of life. I think that is the only path in the next decade or 2 decades to such a realization. But the extrasolar astronomy, they just keep getting better, okay? And so the question is, do they have on their horizon the potential to make an observation, however many generations of telescopes need to happen? Is it physically possible that they could see something that actually is compelling, that all of us agree, oh, you did it. You discovered evidence of a living system. Okay, so it's a bigger community, it's more diverse, and I would say most importantly, it's kind of a grassroots community. This is not mostly composed of scientists who grew up in the astrobiology community. It's people coming in from outside. Okay, so let's, how are we doing for time? Probably horribly since we started late. Very, very late. I'm gonna wrap up as fast as I can, but let's look at some highlights of things that are going on right now. So the part of extrasolar astronomers, of course, they're mostly motivated by discovery and planetary science, and just generally, okay? So they have interests other than these problems. But when they discuss these problems, they're focused on 2 major issues that have been with us since the astrobiology era. Habitability, which people mostly take to mean the stability of liquid water, the surface temperatures in the range where we think terrestrial life can live, and the absence of mitigating factors like a really harsh stellar environment. Biosignatures, I would say, are the biggest issue. So there is a focused body of work searching for biosignatures. In a way, it's the most visible aspect of the latest data coming out of the study of planetary atmospheres from JWST. What is it that they are looking for? They're looking for methane, okay? That's probably the easiest thing to spot in the presence of an oxidized gas, like CO2, oxygen, or ozone. That's evidence of a chemical disequilibrium, something able to generate methane at high rates, even in the presence of an oxidizing atmosphere. Abundant oxidized constituents like oxygen or N2O are taken by themselves as evidence potentially of metabolism. Unfortunately, JWST can't really see oxygen, ozone, or CO. Those will have to wait for the extremely large telescopes, like the 30-meter telescopes. So really, this is our best case. Can we spot CO2 and methane anywhere? And then always keeping an eye out for home run gases. Like, can I find chlorofluorocarbons like we had in our atmosphere until they were regulated out of production? Can I find methyl chloride or dimethyl sulfide, things that are metabolites that are generated in Earth's biosphere? So a lot of this work is being done on M-type stars, which is problematic. We don't have time, so I'm not going to go through this as a side point. The important question is, what are these biomarkers that we're searching for right now with telescopes? One type of biomarker looks for a molecule that only has biological sources in natural chemistry that are recognized. Phosphine is an example. Another type of molecule is associated with metabolism, but many non-biological sources are recognized. Methane is a good example. And then there are compounds that are not themselves biomolecules, but they are products of the breakdown of biomolecules. Okay, so this is the best reconstruction of a molecular atmosphere, according to Heather Knudsen, whenever I want to know what's going on here, I'll go knock on her door at Caltech and ask her, what's the best atmospheric reconstruction anybody's done? She said, this is it. This is the best one that you will find. And it's basically a detection of CO2 in a hydrogen-rich atmosphere and of a super Earth-sized body that's a little too close in and hot to be habitable. So this gives you a sense of what are the sorts of bodies that we're able to see well and what do we see on them. The closest that we've come to a putative biosignature from this work so far are the so-called Hycean worlds. So this is basically taking the concept of habitability and stretching it, saying instead of looking for an Earth-type world that has stable oceans, it's just in the right location to have water stable at the surface, we will look at an almost Neptune-sized world with a hydrogen-rich atmosphere, where the greenhouse warming from the atmosphere allows there to be liquid water stable beneath that deep, thick atmosphere. So such an object has had a decent spectral return that showed methane and CO2 present. That is the biosignature that is plausibly seeable using JWST, and then suggestions of dimethyl sulfide and methyl chloride. So in principle, we did it, okay? We looked, and in the first year or two of possible signatures, there it is. But the problem is the uniqueness argument. So you can plausibly argue that exactly the same chemistry can be part of a dense atmosphere with no liquid ocean beneath it, and the dredging up of thermochemically equilibrated gas from deep in a hot atmosphere. And that these other observations of biogases are just not competent returns. Okay, phosphine. So phosphine is another unique biomolecule seen in the atmosphere of Venus. This one has been pushed back against, some people have argued they can imagine scenarios of making this non-biologically. More importantly, it hasn't been replicated. Okay, you look again, reprocess the data, with independent experiments, it goes away. Okay, and then finally, there is a residual bit of silliness going on, even today. Okay, even with this more serious community, we have the more understandable of them is the attribution of an extra solar system object that flew through the solar system. I don't want to say the name out loud because I'll mispronounce it, but you all remember the story. That passed on a hyperbolic path, or it's in the process of passing on a hyperbolic path through the solar system, this tumbling cigar-shaped object that is experiencing a non-gravitational acceleration, and it was argued maybe it's a light sail, like an alien light sail. The counter-argument is exactly the same thing would happen if you outgassed hydrogen from an icy body very slowly. So maybe it's just a kind of dead comet. A more problematic case is a recent story, coincidentally from the same person, reporting spherules dredged up from the Pacific Ocean floor that are from a meteorite fall that he located from seismic records in New Guinea. and then analyzed and found are enriched in barium and uranium and lanthanum, with a profile that says this must be a technological object. This is basically a spaceship that broke up in the atmosphere. The seismic evidence was actually a delivery truck. Okay, this is like conclusively proven. The seismic evidence of the meteorite fall was a delivery truck delivering supplies to a hospital on the North Shore.
Delivering from outside space.
No, I'm sorry. Okay, you have it your way, I'll have it my way. Okay, the objects were analyzed for their iron isotope compositions. They have exactly the iron isotope compositions of solar system objects. This is in the paper. Like, they actually reported those data, but didn't interpret them in the most obvious way. And the anomalies in trace metals, as soon as anybody who works on fly ash and coal saw them, they're like, oh, that's exactly like fly ash from coal properties. So it's nothing. It's a non-finding. So this is a case where I would put this in the same category with our isonic DNA. The fact that a wrong interpretation was offered, so what? This is science. People do this sort of stuff all the time. But back off when you realize it's dumb, right? Okay? To keep this field from turning silly, this stuff has to get your thumb put on. This has to be stopped. At least the pushing it after it's clearly wrong. Okay, so taking the whole fields as together, there's been a really transformational change in the seriousness of it, the breadth of disciplines involved, but we are still clearly far short of the kind of breakthrough that people are looking for. There's no transformational breakthrough in our ability to observe and interpret biosignatures beyond the Earth. We have no transformational change in our ability to characterize the transition from prebiotic to living chemistry. And the prebiotic chemists and synthetic biologists, they haven't really put together a scenario that lets them go all the way from the precursors to a living system. So there's something, we've made these big advances, we are missing some key conceptual and observational tools that are not letting us make observations or propose observations that will actually be definitive, or conduct experiments that will actually be definitive. So we need some sort of progress. We're out of time, so I'm gonna wind up. But let me tell you what I am doing. So what I am doing to attempt to push forward in the little corner of this area that I work in is to solve the problem of the interpretation of organic remains in samples. And this is really motivated by the Mars Sample Return Mission, which I regard as the only chance to get back a sample where you could plausibly do something. The main problems are, we know multiple sources of these molecules that we have to somehow deconvolve. And we know from looking at gas chromatography mass spectra of rocks from the surface of Mars from the Curiosity rover, the organics that are there are clearly reaction products of breakdown chemistry. So somehow, whatever organics were there, whatever their sources are, they have undergone secondary processing. We have to understand that, sort of correct for it. And so this is the shape of the problem we have to solve. We have to somehow take a sample, obviously correct for anything we do to it by accident, contaminating it, understand how it was transformed in its residence at the Martian surface, or whatever other planetary body we might sample in the future, and then deconvolve the deposited mixture, and only then are you ready to interpret the origin of any putative biomolecules. So how in the world are you going to do this? My answer to it is looking at the isotopic structure, not in the relatively simple ways I talked about yesterday if you were at that seminar, where we added one or two variables to a normal isotopic characterization to extract one piece of information like temperature. Let's go for the whole enchilada, the full isotopic structures of molecules. What would that look like? Even something relatively simple like sucrose has single substitutions that can be in different non-equivalent positions in the molecule. Multiple substitutions that can form bonds with each other that have this characteristic of enhanced stability that I discussed yesterday. Non-adjacent substitutions that aren't really interacting with each other, but these species serve as a kind of pattern recognition signature for the way the molecule was assembled from its precursors. Taken all together, there's like 10 to the 15 versions of this molecule, every one of which are unique. And obviously, if you could observe them all, you have access then to an enormously distinctive signature. Okay, so how do you access this? There's a whole bunch of technologies that
That is under conventional conditions, or that's also variation of pressure and temperature under which those substitutions?
those species will always be present. So any sample of even a simple molecule like an amino acid, it will always exist as a mixture of thousands of isotopic forms. Most of those forms are the multiply substituted forms in their various configurational versions, okay? And so each one of them is a unique chemical species in some sense. It has a unique stability, physical properties, a unique probability of formation from its precursors. And so you should think of there being a composition space that consists of the entire family of orthogonal dimensions that are the amount of each of these unique isotopic forms. How in the world would you observe it? It seems like ridiculous. How could you possibly observe this? The key advance in the capacity to do this came from my lab's adaptation of Fourier transform mass spectrometry to this problem, where we basically take molecules, ionize them, fragment them into pieces, collect subsets of them, and put them in a Fourier transform mass spectrometer cavity. So this is basically a cavity that confines the ions, either magnetically or electrostatically, and excites orbits of them, and then relaxes the excitation and observes remotely, like with an antenna basically, the periods of their movement. And so the raw signal you get from the antenna is the transient, the variation in current that's excited in the antenna as the ions circulate versus time. A fast Fourier transform on that gives you frequency, and the frequency of motion is a function of mass. And so what's exciting about this for our purposes is the mass resolutions reach into the millions. That means we can fully separate the isotopic fine structure of these molecules, observing every isotopic stoichiometry of all the different fragments. And because there are no concretely positioned detectors, you are always simultaneously observing many species at once. This was basically a non-quantitative technology until recently, because the mode of detection isn't really amenable to precise observations. That's the thing we put a leash on, is figuring out how to retrieve a precise data stream from that sort of detector. The basic measurement concept is you take your molecule, break it into pieces. For every piece, there exists a family of peaks that reflect multiple stoichiometries of isotopic substitution. And if you do this at high resolution, every one of those families is deconvolved into an individual isotopic substitution scheme. Pull them all back together, feed them into a matrix algebra scheme that relates them back to their parent positions, and away you go. So what's the furthest that we've gotten? I'll come back to this in a second. What's the furthest we've gotten with this? Here's a simple amino acid, methionine. This is the matrix of all of the mass spectral features that we observed for it. The rows are individual families of peaks, each of which corresponds to a known portion of the molecule. The columns are isotopic stoichiometries observed for each peak. The total family is close to 150 independent constraints on the isotopic structure of that one thing. And then that data can either be dealt with raw or fed forward into machine learning based algorithms for forensically parsing and things like that. Okay, so the concept, this will be my last, I'll like skip over all the crazy stuff we're doing on meteorites because people are had enough, I'm sure. The concept is you characterize in some composition space the compositions of known biological sources, the compositions of known non-biological sources, and the vectors associated with destructive processes that you recognize, like irradiation, UV, exposure to perchlorate, whatever. You then go and collect a substance from Mars and you recognize in it the signature of these taphonomic or preservation-associated changes, and reconstruct the isotopic structure as it would have existed before its breakdown on the surface. If that thing looks like this, you're done. It's abiological. It came from a meteorite. If it looks like this, you go yell at whoever's working in the lab, you put a thumbprint on it, try again. If it looks like this, something you've never seen before, now the game begins. What are my inferences? What's my inductive reasoning tell me about plausible signatures of metabolism that could have led to what I see? When depicted in this way, cartoonishly, you might think, yeah, but I can think of all kinds of non-uniqueness issues. Yeah, but we're going to do it in hundreds of dimensions simultaneously. And that is where bringing in a complexity of observation and nuance, specificity, I think makes this a potentially solvable problem. Okay, I'm going to stop there. This then is a map of all of the other, I think, first order challenges that these different subdisciplines are facing. Okay, thanks for your attention.
So how well does your method work so far?
How well?
Yeah, or I mean, what have
Okay.
you found from this so
Okay,
far?
I'm glad you asked, because all that stuff I skipped over is what we actually did for what I do for a living.
I should have asked.
Okay, so our training wheels for this were not meteorites. Meteorites are tough because there's so many things you don't know about them. The training wheels were petroleum basins because you know the precursors. They're basically breakdown products of plants and algae and stuff. And you have a lot of constraint over the environmental conditions that they experience. And so you have a way of reconstructing what are the specific chemical operations that take me from the biomass to the thing that I observe. First, we're looking at taking solids and extracting from them chemical moieties that we know are reactive in the subsurface, and looking at the isotopic enrichments of the residues. Then we go and look at the products like methane that's coming off of these materials as they thermally mature, and we see the isotopic signatures, mostly as deficits in the multiple substitutions, of a mechanism of creating radical intermediates and capping them. And then we go to the more thermally mature gases and oils and things, and we look at these signatures of cracking, of breakdown of biomass, approach an equilibrium composition. And that's evidence of a kind of metathetic gardening or cycling of these gas constituents at high temperature. So this just gives you a sense that, yeah, it's complicated, but when you bring to bear a variety of constraints on a variety of compounds, you can reconstruct atomistically what is occurring when these biomaterials undergo transformation. After you've done that, you are able to search for isotopic signatures that are unique to synthesis pathways. So this is just like a little visual summary of some of our site-specific carbon isotope data in amino acids from primitive meteorites and from the Ryugu asteroid. And you can see enrichments of individual positions, which when combined with, in some molecules but not others, and then we combine that with a big database of prior molecule average measurements, and we used it to reconstruct a reaction network for the synthesis of these things. The details are like not so interesting, but the important point is this chemistry is not cyclical. This is basically a bunch of precursors going kablooey and making all kinds of products. That's not how metabolism works, okay? Metabolism works by cycling, reusing molecules over and over again, which would lead to a different isotopic signature. And then finally, here's sort of a fun example from a recent paper where we're turning and looking at the aromatic reduced fraction in the Ryugu asteroid and asking, are the polycyclic aromatic hydrocarbons the products of a high temperature chemistry, like a soot generating chemistry that occurs around carbon-rich stars, or of a low temperature chemistry like ion radical or ice grain chemistry in the interstellar medium or the interiors of dense clouds. And the basic tool we're using is to hunt for extraordinary enrichments in multiple substitutions. Many, many, many times the strengths of them that occur at like 300 Kelvin. Chemistry, those are sorts of enrichments that only occur at temperatures of tens of Kelvin, where the vibrational isotope effects get radically amplified at low temperature. And sure enough, this is what we see in the isotopic signatures of a subset of the PAHs, especially the 2 and 4 ring PAHs show these effects, and they go along with deuteration. So this just gives you like a sampler of the ability to go in and make atomistic specific conclusions based upon isotopic structures of different organisms.
Sorry, what is the sign of the results?
Yeah,
They
so-
form in molecular clouds or in stellar envelopes?
Molecular clouds.
Yeah.
Yeah.
Great.
Which is, it is an unsatisfying answer in one sense, because there's a large literature that says, well, probably they're from stellar envelopes, because those are environments that we know have acetylene in them, they could easily generate PAHs. It's satisfying in the sense that the destruction mechanisms in the interstellar medium seem to be faster than can be resupplied by the carbon-rich stars that supposedly provide them. So this is, it means what it means. It says they carry a signature of low temperature, but specific mechanisms that are responsible for that chemistry, if that's true, if it's the right interpretation, that's unclear.